Hello, my name is Raymond Nava, and I'll be talking to you about mitosis. What is mitosis? Mitosis is a type of cell division in which one cell, the mother, divides to produce two new cells, the daughter cells. They are genetically identical to itself. In the context of the cell cycle, mitosis is a part of the division process in which the DNA of the cell's nucleus is split into two equal sets of chromosomes. The goal or main purpose of mitosis is to make sure that each daughter cell gets a perfect full set of chromosomes. This cell is in interphase and has already copied its DNA, so the chromosomes in the nucleus each consist of two connected copies called sister chromatin. Chromosomes are long, stringy, decondensed forms. This animal cell has already made copies of its centrosomes, so there are two sets of centrosomes. In early prophase, the cell starts to break down some structures, setting the stage for division of the chromosomes. The chromosomes start to condense and the meiotic spindle begins to form. The spindle is a structure made from microtubulars, strong fibers that are part of the cell's skeleton. Its job is to organize the chromosomes and move them around during mitosis. The spindle grows between the centrosomes as they move apart. In prometaphase, the meiotic spindle begins to capture and organize the chromosomes, which are now fully condensed. The meiotic spindle grows more, and some of the meiotic tubulars start to capture some chromosomes.
Hello, this is Howard, and I'll be explaining how the metaphase stage works in mitosis. In the metaphase stage, the chromosomes are all lined up in the middle of the cell and aligned at the metaphase plate. The two kinetochores of each chromosome should be attached to microtubulars from opposite spindle poles. Before proceeding to anaphase, the spindle checkpoint and helps ensure that the sister chromatids will split evenly between the two daughter cells when they separate in the next step. If a chromosome is not properly aligned or attached, the cell will halt division until the problem is fixed. During anaphase, sister chromatids separate and the now daughter chromosomes move to the opposite side of the cell. Anaphase begins when the duplicated centromere of each pair of sister chromatids separate, and the now daughter chromosomes begin moving towards the opposite poles of the cell due to the action of the spindle.
I'm Karen and I'll be talking about the telophase. In the telophase, the cell is nearly done dividing and it starts to reestablish its normal structures. This process is what is called cytokinesis. So the two nuclei from each separate chromosomes form and the chromosomes begin to decondense and return to their stringy form. As I mentioned in the telophase stage, cytokinesis is the division of cytoplasm to form two. It may start in the anaphase or the telophase. It all depends on the cell, and it finishes shortly after the telophase. In animal cells, cytokinesis is a contrail, pinching the cell in two. The drawstring is a band of filaments made up of a protein called actin, and the pinch crease is known as a cleavage furrow. Plant cells can't be divided like this because they have a cell wall and are too stiff. Instead, a structure called the cell plate forms down the middle of the cell, splitting the two daughter cells separated by a new wall. When cytokinesis finishes, we end up with two new cells, each with a complete set of chromosomes identical to those of the mother cell. The daughter cells can now undergo mitosis themselves, and it just becomes a repeating cycle.